Traditional taxonomy organized things by how closely they basically resembled one another. Now, this wasn't bad per se. I mean, it made plenty of sense, but wait a second. Things have this habit of changing over time. In fact, even Linnaeus himself realized that, hey, if we plant these different plants in different conditions, they will have adaptations to local conditions. So obviously, they change from our originally described characteristics over time. Now, he and a number of later folks came up with various mechanisms for possibilities of accumulating change over time. But then this guy named Charles Darwin came along, and with some prodding and prompting from a certain Alfred Russel Wallace, he eventually proposed the theory, and I mean scientific theory as in supported by evidence, by plenty of evidence, and not disproved, the theory of evolution. So his ideas were very, very simple. And did you know that one of his friends remarked upon seeing his ideas? How very stupid not to have thought of that. So, what were his ideas like? Well, they had a few requirements. 1. Traits heritable. So traits could be passed between generations. 2. Traits vary. So not everybody in the population is exactly the same. I mean, this is pretty obvious if you've ever gone into a public space with more than one person in there, as in yourself, and looked around. 3. Traits influence survival reproduction. Okay, this is pretty obvious. I mean, we've probably all heard of traits like, say, deformities, yes? Or mental issues? Well, obviously they will influence survival or reproduction, or survival to reproduce. Four. Not an infinitely huge population can be sustained. Now this probably sounds obvious. There is not an unlimited amount of space or resources. So given these 4 things, it is very obvious then that the traits that are best suited to the environment will survive and reproduce better than traits that are not suited to the environment. The result is that these traits become more common in this population over time. Now, say there's another population somewhere else that has different traits that are favored. Well, then these 2 populations will eventually accumulate more and more differences, and one day, when they encounter each other again, they will not recognize each other as mates, or they will be just unable to reproduce, whether mechanically or chemically or whatever. So they have become separate species. Now, during the process of speciation, if we bring them together again, into close proximity again, there is a possibility that they might just merge back into one population, if they haven't accumulated enough differences. They could also go on parallel paths with hybrids continuing to be produced, but not being favored enough to actually merge, and not being disfavored enough to completely diverge. So hybrid zones, as we call them, have a number of different possibilities, and there are examples where they manage to stably continue producing hybrids over a long period of time. But those will be talked about at another point. Now, phylogeny is a study of evolutionary relatedness between and among species. This is related to taxonomy, but not quite the same. So a phylogenetic tree depicts the evolutionary relationships between different species or groups. Now, a clade is a taxonomic group that includes a single common ancestor and all of its descendants. Traditionally, taxonomy had this happening. So what happens when we draw out the tree? Well, we have this, and then these two join together and branch off, and mammals branch off before that, all from a common four-limbed ancestor. Now, there's a problem here. We notice that if we consider a clade a monophyletic group, including a common ancestor, if it's still around, and all of its descendant species, hey, wait, we're not including birds in reptilia, in reptiles. So obviously we can't make a monophyletic clade. With phylogeny, we're kind of obsessed with monophyletic clades, which means that for this diagram, we would have something like this instead. So the reptile clade would include birds. The archosaur clade would include birds. Sometimes this is written without the AE. But we notice that neither of these clades includes mammals. In this case, the mammal clade is the out group, whereas this is the in group if we're going to run any genetic comparisons. The out group should always be diverged further back in time than any of the in group are diverged from one another, because, the in group completely diverged from the out group at some point. Now, in summary, modern biological classification is based on phylogeny, the evolutionary relatedness of species. Now, how can we determine this? Well, we can determine this by fossil record. Oh, we're spotting a pretty smooth trend here in terms of fossil horses going along their various branches, because there are various lineages of fossil horses. And only one branch of the huge tree of various horse species, practically, descended from Hyracotherium, managed to survive to the present day. Now, a clade is a taxonomic group that includes all the descendants of a common ancestor. Now, earlier I said, as few branch points as possible. You may have noticed there's a little bit of a problem with that. Every time you make a branch point, you add one branch to the final total, which means that inevitably you're going to have the same number of branch points, yeah? But if you have, say, genetic sequences, then the as few changes as possible stacks up. Because say, we have 4 species of birds. So A, B, C, and D. Suppose A has A here, and these 3 all have C here. Now, it could be that A just acquired this mutation on its own. But you could also argue that A is the first lineage to spread out. Now, how can you figure out the truth? So A could be here or A could be here. Now, A could also be in between. How do we know this? Because say you labeled the mutation as happening here. Well, then that would solve the issue, yeah? What if, though, you had a longer sequence? and you observe the differences between the sequences, and you make your computer run the analysis to arrange it so that you need as few mutations as possible to get these sequences. That will spit you out a phylogenetic tree that should work well enough. Now, you may have some diverging from one another, such as, say, like this, instead of all coming off one chain, but, well, that's fine. Now, both traditional and phylogenetic classification systems have their advantages and disadvantages. For example, if, say, one of these species, say you're dealing with a bunch of fungal species instead, traditionally they can be a little bit hard because, all we have to go off of is morphology if we can't observe them reproducing. If. That's a big if there, though. Bacteria, well, we also only can really go off of morphology. Now, what about the advantages of traditional classification systems? They have this tendency to be faster, even though they also are a little bit more prone to mistakes. They tend to be cheaper. Phylogenetic classification, you tend to require a bit more processing power. You need to spend the money to get samples and then, well, map out their genomes, or at least part of their genomes. There's a bit of a problem with this. How are we supposed to map out the genomes of dinosaurs? They're fossils. So clearly, sometimes we have to resort to traditional, or not even quite traditional, more like morphological phylogeny instead of actual phylogenetic classification. Well, that's it for this section, and we'll see each other again then next section.